According to the Center for Disease Control, cancer is the second leading cause of death in the United States. Yet scientists are far from understanding how this disease afflicts so many of us. Researchers at the Johns Hopkins Institute for Nanobiotechnology are finding new ways to improve the health outcomes of patients worldwide. Combining talents from researchers working in the schools of medicine, engineering, and sciences, they are unraveling the fundamental mechanisms by which cancers develop and spread throughout the body. By synergizing the capabilities of molecular cell biology with engineering analysis of complex systems to understand the ways in which disease states arise at the cellular level. Normally within the body, cells renew at slow, controlled rates in response to age or injury. Cellular processes are organized, coordinated, and follow the precise instructions found in the genetic information of the cells, known as DNA. The DNA is housed in the cell's nucleus and ensures things are running smoothly. When DNA becomes damaged, as in cancer, cellular instructions become faulty, resulting in the uncontrolled division and spread of cells. These uncontrolled processes begin to harm nearby healthy cells by taking away their critical nutrient and oxygen supply until eventually they exhaust and die. Understanding how cellular signaling becomes faulty in cancer cells is a crucial step towards better treatments. Researchers at Kalina Hrstrova's lab in the Department of Materials Science and Engineering are studying a specific class of membrane proteins that act as gatekeepers called receptor tyrosine kinases, aka RTKs. These RTKs have a unique structure containing an extracellular domain, a transmembrane region, and a cytosolic domain for transmitting signals from the outside to the inside of the cell, thereby regulating the downstream signaling cascades within a cell. To study these RTKs, a chemical technique known as fluorescence resonance energy transfer, FRET, is being combined with high-resolution imaging techniques to visualize and quantify their interactions, thus giving information about the thermodynamically stable states of RTKs under normal versus diseased cells. Another challenge in treating cancer is finding effective and affordable cancer treatments. Current therapies available either temporarily are effective or are nonspecific in nature, attacking both normal and cancer cells. These off-target effects often manifest themselves as the reduction in quality of life with feelings of fatigue, anemia, and infection that follow chemotherapy. To reduce these side effects, a new targeted approach showing promising results is the use of monoclonal antibodies to prompt the immune system of the body to only kill the cancer cells. These monoclonal antibodies, or mAbs, recognize and attach themselves to antigens found on the surface of cancer cells. Once these mAbs attach, they recruit killer B cells of the immune system to release chemicals that destroy the cells containing the antigen. Industrial-scale production of mAbs is done using mammalian cell cultures. The food for these cultures is cell media, which contains all of the necessary nutrients required for their growth. During the large-scale production of mAbs, thousands of liters of media are used in the bioreactor to produce only a few grams of protein product. This low yield is one of the reasons that these mAb therapies are costly to produce. In the Department of Chemical and Biomolecular Engineering, Michael Beatenbaugh's research group is developing an entirely new cell line which works like a factory to only require minimal nutrient inputs from the cell media to produce mAbs on a large scale. By using the biochemical framework already present in prokaryotes, researchers are trying to expand the biosynthetic regime of a mammalian cell. Once a cell line starts generating all the necessary nutrients with the help of its powerful engineered machinery, they don't have to supplement them in the culture medium. Using these tailor-made cell lines, the drugs selectively targeting cancer cells become more affordable. Scientists are using this better understanding of cell machinery to program directed cellular responses into living organisms. Combined with engineering drugs that are more effective and cheaper to produce, allows for targeting diseases at the molecular level in unprecedented ways. Ultimately defeating cancer at earlier stages by knowing exactly how it transforms and manipulates normal cells to become cancerous. These results have demonstrated that the close united approach reflected in the interdisciplinary nature of Hopkins' IMBT program allows new drugs and treatments developed in the laboratory to be quickly transferred into the clinical setting, thereby offering patients access to constantly improved therapeutic options.